Hemoglobin is one of the components in our red blood cell which functions to transport the oxygen throughout our body. So today, we will demonstrate to you on one of the methods to measure the content of hemoglobin concentrations in our blood by using cyanide hemoglobin method using Drapkin solutions. So ferrous cyanides contained in the Drapkin solutions will combine with the hemoglobin inside our blood and will form a stable complex named cyanide hemoglobin that can be measured by using calorimeter at a wavelength of 540 nanometer. The reference range for hemoglobin concentrations for male is 13.5 to 17.5 gram per deciliter. Meanwhile, for female is 11.5 until 15.5 gram per deciliter. Before we proceed with our demonstrations, we have to make sure that our apparatus is complete and ready. So here we have freshly prepared Drapkin solutions, clean wipes tissue, clean glass test tube with test tube racks, sample in EDTA tubes, clean cuvette with cuvette racks, adjustable micropipette with 20 until 100 microliter, micropipette tips, vortex, and calorimeter with 540 nanometer filter.
So in this video, I'm going to share with you the sample preparation for the Hb quantitation. So we are going to use the Drapkin solution as our working reagent. Pipette 5 ml of Drapkin solution into a clean glass slide. Then transfer 20 microliters of your sample into the pre-pipette Drapkin solution. Make sure you mix your sample before you do the pipetting by inverting 3 to 5 times. Transfer 20 microliters of your sample into the Drapkin solution pipette. Just a gentle reminder, make sure you wipe off any excess sample from the pipette tips. This is to make sure that you are dispensing only 20 microliters into the Drapkin solution. You may use a vortex to help you to make sure that your sample is mixed. Leave your sample to rest for at least 2 minutes before you can read it with the calorimeter in a dark room. While waiting for your sample to be ready, so hereby I'm going to share with you the hemoglobin standard set. You can buy it commercially. This is the kit. We have already known the concentration. It's 20 gram per deciliter. We are going to read the absorbance of this hemoglobin standard of the non-concentration of the 20 gram per deciliter and substitute it into the formula given after you have read your absorbance for the unknown sample. So from there, we are going to get your concentration of your unknown sample.
And now it's time for us to measure the absorbance of the unknown sample and the absorbance of the hemoglobin standard. We are going to transfer 1 ml of drug-in solutions into this clean cuvette. The reason why we are using drug-in solutions as our blend is because in this practical, drug-in solutions is our working reagent. Now, we are going to transfer this unknown sample into the clean cuvette. But before that, we have to mix the sample by using vortex to make sure that it is well mixed. And now it is time for us to prepare the cuvette for hemoglobin standards. But before that, gently invert this bottle a few times to make sure that the solution is well mixed. For your informations, the types of cuvette that we are using have two types of surfaces. One with the grooved surfaces and another one with a smooth surfaces. Please make sure that you wipe the surfaces before you put it inside the chamber. And also, please make sure that you only use smooth surfaces facing the light source before placing it into the chamber. Before we proceed with the absorbance measurements of the unknown and known sample, we have to do the zeroing process by using blank cuvettes which contain drug-in solutions. Take the blank cuvette which contain the drug-in solutions. Wipe the smooth surfaces by using clean chem wipes and place it inside the chamber in calorimeter. Press 0 to proceed with the zeroing process. Now we are going to measure the absorbance of the unknown sample. Take our sample cuvette, wipe it with a clean chem wipes, and place it inside the chamber in calorimeter. Record the absorbance that's shown on the calorimeter screen. Now we are going to measure the absorbance for the hemoglobin standard with a known concentrations of 20 gram per deciliter. Wipe the smooth surfaces by using clean chem wipes. Put it inside the chamber and record the reading shown. Then we substitute the value into the formula given to obtain the hemoglobin level for the unknown sample. Nowadays, An automated analyzer is used in a modern lab to help to determine the parameter for a full blood count of a patient for more reliable and faster result.